Good morning. Welcome to 10 Years Hence. Our speaker this morning, Dr. Gill Bergeron, is an associate professor of developmental biology, an associate professor of computer science, and an associate professor of pediatrics at Stanford University. Dr. Bejerano's lab studies genome function in human and related species. The lab focuses on a fundamental question in human genomics: mapping genome sequence to phenotype and extracting specific genetic insights from deep sequencing measurements. The Bejerano lab has an interest in gene cis-regulation and uses its joint affiliation with Stanford Hospital's Pediatrics Department, Stanford's Developmental Biology Department, and Stanford's Computer Science Department to apply a combination of computational and experimental approaches. The lab collects large-scale experimental data, writes computational analysis tools, runs them massively to discover the most exciting testable hypotheses, and experimentally validates the hypotheses. Dr. Bejerano graduated summa cum laude from Hebrew University with a Bachelor of Science degree in physics, mathematics, and computer science. He holds a PhD from Hebrew University in computer science. He is a member of Numenta's Technical Advisory Board, as well as a member of BioX, the Cardiovascular Institute, the Child Health Research Institute, the Stanford Cancer Institute, and the Stanford Neurosciences Institute. Please join me in welcoming Dr. Gill Bergeron.
Okay, can you guys hear me? Maybe? Huh? Now I can hear me. All right, awesome. So good morning, everyone. Appreciate the introduction. It's a pleasure to be here today, not done with you guys. You can see I'm even appreciative, or not, I am, of the weather. I prepared a beard, especially for the occasion, but it's just lovely out there. So it's a pleasure to be here with you today. I thought I'd give you a bit about, let me go stand closer to that machine. A bit about my personal angle for being so happy to get this invitation to come visit Notre Dame. It's my first visit here. So I've been a faculty at Stanford for about 11 years. And as a faculty, I would take 1 or 2 students a year to something that would amount to a 5-year journey together. And out of, let's say, and the lab is not only PhD students, there are postdocs and undergrads, but really from the PhD students with whom typically I spend most of, the longest time together, I already have 3 Notre Dame alumni. Aaron Wenger on your left, Jim Notwell in the middle, both of which have graduated already, and Bo Yu, who's a student in the lab right now. So I was very happy to get the invitation here. I figured, I'll go check the place where these folks come from. So pleasure to be around. The other thing I would say, and that would lead us more into the talk today, is, even for somebody with my background, computer science, medicine, I've been spending more and more time talking with business people, and I'll give you a sense of that today, and hopefully some of the talk you'll hear will be of interest. Okay, so delighted to be here. Appreciate the invitation. All right, so why genomics, right? So first of all, maybe in this context, it's good to notice that the compound annual growth rate of genomics for the foreseeable 5, 7, 10 years, and likely far beyond that, is phenomenal, right? Between 10 and 20%. The market size is estimated in the tens of billions of dollars. So genomics is exploding. I also think that what I'll talk to you today, or I'll try to make it interesting, is in itself, whether I make it interesting or not, you decide, is a great example for the future of medicine, for what happens when artificial intelligence enters a new field, and artificial intelligence is very hot in the marketplace today. So I'll show you an example of AI plus X being medicine. I also think it's a good topic for 10 years hence. So hopefully, I'll tell you where things are now, and I'll try to hint at what the future is. Just try to imagine what 5 to 10 years from now would look like with the systems I will describe to you today. All right, so let's start maybe by defining what is genomics. I'll define genomics very briefly to you in my own words. Let's see that we're on the same page. Okay, so what I tell computer science students, and I always run around between different camps, computer science, the medicine student, I think some of the slides I brought today are a mix of the two. So I tell them, essentially genomics is the study of the most amazing operating system on the planet, and that is the human genome, okay? So why is that? So if you look in the mirror, what you see is essentially 10 to the 13 cells all working together, okay? 10 trillion, I think that ends up with. These 10 trillion cells all started out as one cell, one fertilized egg, and that one fertilized egg, half of your genome came from mom, half of your genome came from dad, and a sprinkle that is purely yours. And then that one cell becomes 2 cells. Those 2 cells become 4 cells. Those 4 cells become 8 cells, up till you get to 10 trillion. And to a first approximation, in every single one of these cells, there's a copy of your genome. The copy that started out in the first cell, that was you, is handed over to the daughter cells, and their daughter cells, and their daughter cells. So the genome is essentially this operating system that runs every single cell in your body. Conceptually speaking, it's a very long string over a very short alphabet. 3 billion letters long over an alphabet of just 4 nucleotides or bases, A, C, G, and T. Okay, so just remember it. The exact, the first approximation, the same genome runs every cell in your body. Very different cell types, but every cell in your body is run by the same genome. All right, so the biggest challenge in genomics, I would postulate, condition on the challenges we've solved already, is essentially computational. How does this piece of code for that? Us. Code and output, okay? And if you can solve this coding question, it has profound implications for all of our lives. I'll give you some examples. You can look at people's ethnicities. You can appreciate how we've taken, how humans have migrated, patterns of migrations of humans across the planet. You can see extinct hominids and see how they've contributed to the gene pools of those of us who are still here today. You can see fascinating patterns from examining the genomes of extant and even extinct hominids. We can study the genomes of related species. Notice that I have in the small print over there, right, all of us humans are 99+% identical at the genomic level. Primates are 90+% identical at the genomic level. So when you just compare the genome, the similarities are striking, both the similarities and the differences. And you'll see I have, computer science language there, right, that you can look at. We can literally go now and find bugs in people's code. These are genomic mutations that lead to genomic or genetic diseases. I'll use the term indistinguishably. We can use our genome to treat people better. So for example, if I want a certain drug to be at a certain level in the cells of your body, we can actually now look at people's genomes and appreciate that different peoples have an operating system, the genome. The cells of different people pump out drugs at different rates. So I can give you a higher dosage or a lower dosage based on how quickly your cells will pump it out if I want a certain level in your cells. So we can do this patching, right? I'm not treating your genome, but I'm using your genome to give you a more focused, adapted drug. Now, we also essentially are doing today what I can call software verification. So if a couple is referred to the in vitro fertilization clinic, we can now essentially take sperm and egg, put them together in a dish, get to the point where we have 50 viable, let's say, offsprings, potential offsprings on the dish, we can wait for each of these to grow from the 1 cell to the about 100 cell stage. At that point, you can literally come in with a very fancy straw and slop, I believe is the technical term, slop off a handful of cells from any embryo you like. The embryo would continue to grow perfectly normal. Take those handful of cells, sequence them, and choose which embryos you would like to implant and which embryos you wouldn't like to implant. So that's obviously often done in cases where we know some bad mutations lurk, and we would like to make sure that the offspring does not inherit those. So we can literally do software verification now, essentially ask, should this genome be booted up, or shouldn't it? And last but not least, we are actually become capable of fixing genomes. We can now go to this string of 3 billion bases, and if you like, again, to a first approximation, change any base you want. An A to a T, a C to a CCG, drop a few letters, anything you can do in a word processor, let's say, we could do to a genome. What we really need, and I hope I use these words carefully, what we really have today is something like this trinity of the fact that reading genome, I'll show you numbers in a minute, has become extremely cheap. So we can read as many genomes as you like. Everybody in this room, anybody and everybody. We can change genomes into almost anything. And all we're missing there to kind of complete this trinity is we need to understand what it is we're actually doing. What is it that we're reading? And more importantly, what is it that we would like to change? And today I'll tell you a bit about mostly the stuff of understanding, okay? All right, so let me tell you briefly what we do in genomics before we move on to describe the field. We do essentially 2 things. This is maybe the technical side, or I'll skip that. We build systems that can diagnose millions of people. This is what we'll talk about today. And we apply those systems to real people so we can diagnose real patients and get, for example, real thank you letters that really warm up our heart and make us feel like we're on the right path. We study amazing species adaptations, not just humans. If you look around other mammals, for example, you see amazing adaptations. For example, in the way bats and whales can echolocate to catch their prey. Well, it turns out that bats and whales also adapt their genome the same way, in some cases, to kind of hone in on these new capabilities. So we can see those independent signatures in their genomes, those and many others that we study in the lab. And last but not least, we try to understand gene regulation. This is a technical topic that I don't think is much of interest today. If you're more from the bio side, if you're familiar with the bio side, essentially what we do there, well, I guess the analogy is a mix of bio and computers. We try to look at the runtime stack of your genome. What is it doing in some context? And try to see the logic from the stack. And in computers, that would be looking at the actual machine code and what's happening there and trying to understand the program that is run behind the scenes. Okay. So I think I mentioned all that. Maybe I'll just mention, we've been doing genomics for a long time, since 2003. And I think everything here is just to tell you that we have some pretty, we've been able to publish in respective venues, Nature Science, Nature Biotech, Nature Genetics, et cetera, both in the context of genome evolution, genome function, and genome disease. And that just comes to say, maybe what I say makes sense. All right, so let's finish with that. I think the last thing I wanted to put, although it's been beautifully introduced as well, is something that you shouldn't think of it. Well, you know what? Let me give you the introduction first, and then I'll tell you why I tell you about it. So let me tell you about my own career trajectory very, very briefly, right? Started out in math and physics, added computer science, still in my undergrad, really enjoyed it. Took a year off, came back, wrapped all 3 of them. You have a lot of energies after a year off, maybe too much. Then I moved into a specific field of computer science, machine learning. In it, I tried to apply my tools into computational biology, and only later, really, did I become a genomicist. So then I started working on comparing the genomes of different species, looking at functional genomics, and more recently, Evo-Devo is something we won't talk much about, but got into human disease. So the point is, the only reason I show this trajectory is I think it's a typical trajectory for many people like myself, in the sense that we deal with human patient genomes, but we come from the quantitative sciences. Okay, just look at where that trajectory started versus where it's at now. I am not a doctor in the medical sense. And technically speaking, I didn't even grow up in the biological sciences, although that is all essentially of what I do now. So we'll get to that maybe a bit later. We'll talk about some cultural issues later on. All right, enough for us, right? Let's get to this topic of discussion today. So the main topic of discussion today is patient diagnosis. This is what I'll show you, patient diagnosis from the genome and how that is changing the field of medicine. Where would medicine go in the next 10 years along with these capabilities? What are some of the challenges we have? And I think a lot of potential for wonderful things to be done, perhaps by people in the audience today as well. So let's see. So first of all, let's just say, if you can put your finger on what is it in the patient genome that makes them sick, that is a wonderful thing. Why is that so? It's wonderful for the patient because typically when we know what is it in the patient that's broken, not just the symptoms they present, but what is actually wrong in their genome, we can take better care of them. And for some of them, there's even a first hope for a cure. There's never a hope for a cure if you don't know the root cause. So once you have the root cause, you suddenly have something to aim for. For their families, it's important to understand some of the patients we deal with, the disease manifests itself very early and very severely in very young kids. So these could be the first children of very young families. And one of the first questions that we're asked, or the physicians that we work with are asked, is, should the family have another child, right? What is the likelihood, if I have another child, that they would have the same disease, or what is the likelihood that they would be healthy? Again, if you can put that X on the spot on the child genome, you would know whether that is a spontaneous mutation in the child genome, something that came from mom and dad, in which case you would like to go to the IVF clinic. Without a diagnosis, you cannot make those claims. It's also wonderful for the families to know what lies ahead and to go into very focused support groups of families of other kids who have exactly the same issues as theirs. And then last, but definitely not least, for the clinical caregivers themselves, I'll show you some of the diseases we solve today are rare diseases. These would be, 200 individuals across the country. So your clinician would have never seen another case like the one they're treating now. And if they could talk to somebody in Albuquerque who treats another family like yours, and maybe the kid there is a bit older than yours, they would see where the trajectory is going, what has been tried, what could they do. So a diagnosis is a very, very important thing, okay? And we are making wonderful progress in diagnosis, in genomic diagnosis. We're even starting to cure the uncurable. I'll give you a whiff of that in a second. But, so today I'll show you a few buts. Here's one. We're not doing it in the desired order, okay? So think about what happens. This is a nice example, perhaps, of when you disrupt a field, some of what happens and some of the repercussions. So what do I mean by not doing it in the desired order? First observation is, if you look, for example, at the hospital or just at people who are sick, you would want to treat the most common diseases first. So you would want to go after things like asthma, or you would want to go after things like Crohn, perhaps. Okay, these are diseases that are focused on a single organ. Many people have them, and if you can help all those people, that would be wonderful. This is not how genomic medicine is advancing. Okay? Instead, what is happening is we are solving first the diseases that are simplest in the genome. Except simplest in the genome does not look simplest in the patient. Why is that? If you take a patient genome and you just break it in one tiny place, again, think of the genome as a piece of code. What is beautiful about our genome is that it's in every single cell of my body. So if I just broke one tiny place, one letter was changed, the genome is wonderful at reusing code. So the same code is used in my liver, in my lungs, in my brain, and if the same piece of code I broke, a tiny, tiny change in the genome, it would mess up my liver, my brain, and I forget what was the third tissue I gave you, my spine or whatnot. So you can make very simple bugs in the code that would manifest in a very complicated way in the patient. And we are solving those first. So we're not solving what looks phenotypically simpler in the output. We're solving what looks genotypically simpler in the genome of the patient. And also, as I alluded to, we're not solving the common diseases. We are first and foremost diagnosing very rare diseases. I'll show you in a bit. We diagnose thousands of them, but each of them individually is quite rare. So from a clinician's point of view, if all you want to do is care for patients, there comes this new disrupting technology, and it's not going after the big piece. It goes after, all these patients with exotic diseases that we could never know what to do with them, and there are 5 of them in the state, and that's it. And they solve them. They even cure them. Right? It's not the right order if you're patient-focused. It's the right order, though, if you understand what is going on. You start from the simplest things in the genome, and you'll build your way up to more and more complex things. So this is one thing that the medical establishment has to get used to and is getting used to now through patient advocacy, et cetera, groups of patients, small groups of patients, before you start aggregating them, who have always been in the periphery are being pushed front and center because their genomic underlying cause is simple, even as the manifestation is complex. All right, that's one observation. So these patients that I tell you about, the ones we can bring the most value to today, have usually undergone such a complicated process of diagnosis that it's actually called the Diagnostic Odyssey. So somebody went and collected the stats. So the kids that we treat today would typically take 7 years to get to a diagnosis. 7 years, and they would see up to 5 to 7 specialists, right? So they would be, the first organ system that would fail, they would go to an expert of that organ. But that expert, imagine you went to a cardiologist. A cardiologist is focused on your heart. And then a liver issue came up, so they would be referred to a liver specialist, et cetera, et cetera. You had to wait until somebody said, hold on, too many systems, what if this is genetic? Okay? And then you had to sequence the patient, and sequencing meant different things in the 2000s, in the 1990s, in the 2010s, than it is now. And ultimately, from whatever sequence, whatever glimpse of the patient genome you could get, you had to put your hand on the cause. And it used to be that you couldn't just get all the patient genomes we get today. You had to guess. You'd look at the patient, you'd squint really hard, and you'd be like, it's that piece of the code. Let's go and have a look at it. And if it wasn't that piece of the code, you'd still have no diagnosis. So these patients underwent a long diagnostic odyssey, which we are going to disrupt completely in the next few minutes. All right, here's another but. Genome-based diagnosis precedes our biological understanding. What do I mean by that? There is a lot we already know in biomedicine, a lot. If you just sum the facts, the procedures, everything we know how to do, let's say they're in the millions of useful knowledge points, it's enough to look at our life expectancy and how that has gone up in the recent centuries. So we know a lot, but we have to be very, very cognizant of how much we still don't know. And that is a lot more. Okay, so just imagine this cup. There's a giant cup. It's, 15% full. It keeps filling up. The 15% is a huge amount of knowledge. but you have 85% that we still have to learn, okay? So we know a lot about biology, but there's a lot more that we still don't know. So for example, if you manage to collect 3 individuals, okay, unrelated to each other, all 3 of them seem to suffer from the same symptoms, an issue with the liver, and the same issue in their liver, the same issue in their brain, and the same issue in their spine. If you collect these 3 individuals and you sequence their genome, so first of all, I should say, what these individuals have, you can see it's a disease that affects multiple organs because of code reuse of your genome. So those would be called syndromes. Diseases of multiple organs are called syndromes. And as I said, these diseases used to be extremely mysterious. So they would literally be called Dr. X, Dr. Y syndrome. Widerman-Steiner syndrome. All that says is that doctor, I should say, it's a medical institution, Dr. Widerman and Dr. Steiner were the first to appreciate that multiple people who suffer from the same set of organ diseases probably have an underlying genetic cause, even if they couldn't find the genetic cause to begin with. So if you find 3 patients like that, and you can easily now sequence your genome, you can suddenly say, look, when I take these 3 genomes, I compare them to thousands of healthy genomes that I have now, and there's only one thing these 3 people share, a mutation that seems like a very bad idea that messes up gene X. That is the only thing these 3 people share, and none of thousands of healthy individuals share. You have probably just diagnosed a new disease. And if we have time at the end, I'll actually show you just batches of how this goes. Just take a bunch of patients for the same disease, as little as 2 or 3, sequence their genome and ask, what do they have in common that nobody else that's healthy has? So often enough, we know close to nothing about the gene that causes their disease. Or at least we know nothing about the disease implication of the gene. Maybe we have some biochemical understanding of what it does in the context of some cells. So genomic diagnosis goes ahead of our understanding of biology. That's important to understand. Another thing that's useful, I don't know if you remember it in my first slides, the Dev Bio department have a nice logo where it's like mice and flies and all kinds of organisms running around the logo. Biology has always loved model organisms. We can induce diseases in them. We can cure them. There's a giant effort, for example, to go in the mouse genome and break every single gene separately and just see what diseases come up, okay? Well, it turns out now, suddenly, with the availability of genomes, human has become our best model organism. If we can sequence entire hospitals, we would learn a lot about diseases and their underlying genomic causes. So that's another paradigm shift, really, in biology. So it also turns out, in a very nice way, that you don't have to understand everything in order to cure. So we are now curing effectively the incurable. How are we curing them? We essentially go to their genome, we realize what is broken in their genome, and we give them a life-saving code injection. We put a copy of a function that works next to a copy of a function that is broken in their genome. And for some patients, that is all you need. So here's one of the more dramatic examples that are already in the market today. Right? You take these SCID patients, they're colloquially known as bubble boys, right? Kids who have, whose immune system has not booted up. Okay, the kid otherwise grows mostly normal, but they have no immune system. And when they have no immune system, you essentially have to raise them in these giant bubbles where you filter their air and you sanitize their food and you can touch them only from the outside, et cetera, because they have no immune system. They could not deal with any bugs. These are kids, some of these kids have a very small single bug in their genomic code. You can think of it, there's a whole set of dominoes that just waits for the first domino to fall and then the second domino would fall. All the dominoes would fall and you'd have a beautiful immune systems. These kids have a break in the first domino. The first domino never falls. Their immune system never boots up. Okay? What we can do today is we can take bone marrow from the kid, We don't have to transplant from their brothers or sisters and hope that the body does not reject cells that are not theirs. You can take cells from the kid, inject a working copy of the gene that's missing into those cells, put those cells back into the kid, and the set of dominoes falls, and that's it. This is the exact same kid on, what does that be? On the right there, exact same kid on the left, completely cured, right? Walking around the neighborhood with a stick, right? That's it. You've cured the incurable. You've booted up their immune system. We don't understand the entire process. We have a conceptual understanding of what it is that we wanted to do, and we've been able to do it, right? Again, we don't do it in the desired order. This is exactly the same thing as the diagnosis itself. We can fix simple things in the genome first. Simple in the genome does not mean simple in the phenotype, and it does not mean common in the population. These are very rare cases, but that is where we're starting. And like I said, it's not a desired order. You wish you could cure everybody with Crohn with a similar approach, but it's a logical order. You start with the simplest things genomically, and you work your way up. Now, there's an interesting case here to be made that I will just point to, which is, if you've actually taken a kid like that and you've cured them, they're done, right? It's a wonderful miracle. This is a business school. What is the business model of curing people? Let's think about that, right? So doing these procedures, like I showed you before, is in its infancy in individuals. It costs an arm and a leg to develop these techniques. And then you cure a patient, and they never come back, right? They're healthy, okay? You cure a patient, and they're done. You are done. You've performed a miracle. Now, conversely, if you have a chronically ill individual, you have a customer for life. So, one of the biggest drug companies in the world, GSK, has recently spun off their gene therapy division because revenue would go up for a while and then diminish. Very few people, you touch them once they're cured, how much money can you make off of it, right? And very recently, if you guys follow the news, a Goldman Sachs analyst dared ask the question, is curing people a viable business model? Okay, so I'm on the science side, right? So I'll just make two comments on this. There's a couple silver linings here, right? One is we can treat groups of diseases with the same recipe, the same kind of fix, going to the same types of cells. And I think that should make costs drop down. And it's actually quite clear that society, one way or another, will actually force us to do it. They'll force us to do it or them to do it for ethical reasons, and also for the future benefit of all of us, because we understand that if we know how to solve one bug today, we'll be able to solve 2 bugs tomorrow, 7 bugs after that, and we'll start going after those more common diseases that are simply more complicated in the way they manifest in the genome. But this is something to think about. All right, so let's start thinking more about this 10-year hence model. Let me pick this topic for you that I alluded to before. Think specifically about fixing what we call monogenic diseases. Diseases that are caused by 1 or 2 mutations to a single gene in the patient genome. Let me show you the landscape there, and I think it would be a great example of how biomedicine is getting disrupted and what is happening there. So first of all, if you aggregate all these different groups of patients, the aggregate is large. Okay, single gene diseases affect 5 to 7% of the population. So you're not talking small potatoes anymore. 30 million people in the US alone. 30 million people in Europe. 7 million babies born every year suffer from these severe monogenic diseases. So the market in aggregate is large. The diagnostic rate, if you take all of these monogenic diseases, the The diagnostic rate now is 30%. 30% of people we think suffer from a single disease break. If we sequence their genome, we can say, you know what? It's this thing. It's this in this patient, it's that in that patient, and it's that in that other patient. This rate was 10% 4 or 5 years ago. So we're getting better and better at putting the finger on the diagnosis of different patients, and we can do it for 5,000 different diseases. So this aggregate is a nice market, okay? The machines we have at our disposal are awesome. You can buy one machine that will give you 100 genomes a day. That's now. Wait a year or two, you'll get a machine that can do 1,000 genomes a day. So the machines can just crank through your entire patient pool, anybody, your entire population. We can take what the machines put out and build the genome from that really fast. We can write beautiful code, accelerate it in hardware, like something that we've done recently. Anything that comes off the machine immediately becomes the patient genome, okay? Just go and sequence everybody. We said 30 million people are waiting in the US alone, right? And the production cost is phenomenal. It took us $3 billion, and I forget how many genome centers around the world, maybe 20 of them, to sequence the first human genome in 2001. $3 billion. We can get a patient genome now for around $1,000. From 3 billion to around 1,000 in 15 years. This, as some of my colleagues like to point out, is like taking a Lamborghini and bringing it down to the cost of a dime, just by producing it so much better. That's not too bad, right? We could go and sequence everybody, or at least a whole lot of people, and indeed, these are the plans. If you look at projections for the next 5 to 7 years, the plan is to sequence 60 million people. We have people who need it, we have machines who can do it, and we have costs that we can bear. Let's do it. What's the problem there? What is the problem with that model? Well, the problem is you are generating here a data tsunami, okay? Why is that? You have all these patients, you have all these machines, but humans, at the end of the day, would have to approach these 60 million genomes and put their finger on the diagnosis, okay? All of that data ultimately ends up in a human expert's hands. So this, the quip that goes along with those concerns is you're going to have $1 million interpretation of the $1,000 genome. That effectively means you're going to have to pay experts so much money to figure out this cheap genome of yours that the assay is no longer cheap at all. I won't even focus on that. Let me rewrite this for you. It takes a work week to interpret a single patient genome, to put your finger on the diagnosis. The minute it comes off the machines who work at huge volumes and at low cost, now you need a work week to put your finger on the diagnosis. We don't have 7 million work weeks in the workforce, by far. We just don't have the people who would go and say, in this patient it's that, in that, right? We have 5,000 plus different diagnoses that we can make today. The load on the human expert is unsustainable, okay? So what can help, right? What could you do to help, okay? Well, the obvious answer is, dude, we need a computer, right? Why is a computer a good idea here? It has infinite memory. If you need more memory, just go and buy more memory. That's good if you're trying to diagnose 5,000 plus different diseases. It has infinite stamina, right? A work week for a computer is 24 times 7, right? It could just pump through things, and that's good if you think about 7 million babies a year. 7 million babies a year is a lot. And it has infinite speed, right? The program's not fast enough, just go and buy more CPUs. Just throw more power at it. This is wonderful for places like the neonatal intensive care unit, right? Where you get the most severely sick kids, and if you can immediately throw their genome on a sequencer and immediately get the diagnosis, the genome itself doesn't help you at all. You need a diagnosis. You could treat them and potentially save their lives. So a computer is a wonderful thing to have in this context. It's the only way to drink from this fire hose, I would argue. Problem is, the computer lacks cognitive abilities, right? So these we replace with two things. We replace it with artificial intelligence, which effectively says machine learning, or really, what the example that at least I'll show you today, classifications from examples. We'll show the computers things we like, we'll show the computers things we don't like, and we will tell the computer, here you've seen a few things I like that I tagged manually for you, things that I don't like, go into this entire body of knowledge and get more for me like that. I'll walk you through an example of that in a second. And then at the end of the day, you want the machines to help you out because there's just not enough people to do the analysis by themselves. But really, you have to have human expert supervision at the end of the process, right? So an expert must approve, a human expert must approve any diagnosis that a patient will be treated on for the remainder of their lives. But really, the experts themselves really need the machine as much as the machine, I would argue, to some extent, needs the expert to give it a seal of proof. Right? So, artificial intelligence to the rescue. So there are, if you actually looked at the workflow of genetic medicine as these giant data sets of lots and lots of patients are sweeping through, there are many tasks that can be many repeating tasks that can be automated there. Today, I will focus on one example with you guys. Okay? The one example is as follows, right? Remember, every single gene disease is rare in itself. So if I have a patient with a rare disease, which I think is genetic, I most likely have never seen a patient like that before. And also most likely, that disease has never been discussed in med school, because even though med school is whatever, 4, 6, 7 years, you do not just do an Encyclopedia Britannica over every single genetic disease. There's simply too many of them, right? And the human mind won't necessarily remember all of them. So the way you do it is you diagnose your patient by consulting in the medical literature, okay? Essentially, you sequence your patient, you take the patient genome, compare it to all healthy patients, all healthy individuals who've been sequenced. We now have common repositories of thousands and thousands of healthy individuals. And you say, you know what, my patient has 200 funny-looking things in their genome that I've never seen in a healthy individual. I wonder if one of these could be the cause of their disease. And then you literally go to the biomedical literature and you try to find a paper that triangulates your patient, i.e., find in the literature patients with the same set of phenotypes like your patient. So here are 5 people suffering from the same thing my patient suffers from. And in that paper, they've worked out that those 5 people have a certain mutation to a certain gene. You look in your patient, then they have exactly the same mutation in exactly the same gene. You're like, now I have a diagnosis for my patient. My patient suffers from exactly the same thing as those patients. All I have to do is find that paper, okay? So how do humans approach this? So humans approach this in what we fondly call a Dr. Google approach. In a Dr. Google approach, you take a very busy, highly paid doctor, and you put them in front of Google. And you say, hey, doc, Google away. Is it this gene? Ho-hum. Is it that gene? I don't know. What about this third gene? You have a list of 100 to 300 genes, 5 to 10 patient phenotypes, and you try to match. You try to match from this. This is how it takes a week's work to solve the cases that we can solve. And we can only solve 30% of cases, right? So for every case I look at, I don't even know if I can solve it today because I have a chance of 1 in 3 of actually providing it. This is how humans would approach this. How would you approach this if you were using an artificial intelligence system? Let me show you one system that we built that tries to approach that. And again, just try to get a sense from here of where the future of medicine lies and some of the disruptions that I think are going to wonderfully propagate it forward. So So we call our tool Amelie. I'll show you in a second why. Doesn't matter. This AI tool just goes and reads all the literature, right? It's not that complicated. You just say, hey, computer, no, here are all the papers in the world. Deal with it, right? So what does the computer do? First of all, so the way the biomedical literature is built is titles and abstracts of 26 million papers are available to anybody, okay? So we tell the computer, look, out of these 26 million abstracts, here are 1,000 we like, and here are 1,000 that are not about genetic patients. You just say, this is about genetic patients, that's about genetic patients, and this is like stuff we're not interested in this context. You know, sport injuries, all kinds of other stuff related to biomedicine that is not about patients with single gene diseases. And then you set it loose on all 26 million abstracts. Essentially, think of it as like a human with infinite time on their hands. So now the machine goes through all 26 million abstracts and says, this may be interesting to you, probably not. This may be interesting to you, all 26 million. You get a huge pile of abstracts that could be relevant to any patient with a single gene disease. And then, you know, sitting on the shoulders of giants who have fought a battle with the publishers before us, we can now actually go, every publisher that respects themselves have now an API, a programmatic interface, where you can go, if you've paid their exorbitant fees, you can go and download as many papers as you like. So we take all this pile of, say, 500,000 papers we would like to read, and we get the full text of all 500,000 papers. Again, something a human with infinite time and infinite memory would do. And then we try to extract as many facts as we possibly can from these 500,000 documents. And I won't go into the technical details. Essentially, we just say, look, you know, which gene is broken, how is it broken, what does it cause in the patients. Memorize it across the entire body of literature. Okay, so this is what we do up front. Now, for every single patient you give me, I compare that patient, again, the code compares that patient to the entire body of literature. Okay? So we ask, you know, I can go through every single paper that the machine has read and tell you how good of a fit that paper is for your patient. For example, does the paper even talk about a gene that your patient has a suspicious-looking mutation in it? If it's not relevant. So let's only look at the papers that discuss a disease gene that your patient has a suspicious-looking mutation in it. And if it does, I'll just tell you, you know, in the paper, they say the patients, if you get this mutation, they suffer from A, B, C, and D. Your patient suffers from E, F, G, and H. How similar are E, F, G, and H to A, B, C, and D? So we rank the entire body of literature as to how well it can explain individual patients. We order it by genes, and that is the first time we call the expert into the room. So we call the expert clinician into the room, and we tell them, look, you know, here's the patient, here's what they suffer from. We think this paper has the best chance of giving you a diagnosis. Why don't you look at that? And the expert looks and says, ideally, dude, the first paper you gave me, that's it. That is the diagnosis for my patient. If it's not the first paper, maybe it's the second paper we give them. Maybe it's the third. We just rank all the literature in the hope of solving something. Okay? So, and like I said, ideally, you want this to be, the first paper you show the expert would just be a slam dunk. So let me show you some test results that we've done on 200 plus patients. If you take 200 patients with a known genetic disease, our system, this Amelie system, can, for 2/3 of these patients, we can place a paper that diagnoses these patients in the first position, i.e., the minute a clinician comes and looks at this case, we show them the first paper we show them solves the patient. If you're willing to look at the second gene and not just the first, maybe the top gene we picked is not the right one. If you look at the second one, it's 70-something. If you look at the third one, it's 80. And if you're willing to read only on the first 8 genes, we will solve 90% of cases for you. So if you tomorrow get 1,000 cases off the machine, you come in the morning and there's 1,000 cases, right? So today, if I was, I'll say it cautiously. If I came into work today and I did not have these tools and somebody gave me 1,000 cases, I would quit. I would be like, dude, you know, Impossible, right? That's like a 5-year workload, and you're going to give me another 1,000 tomorrow or next week? No way, right? With these tools, you can order the 1,000 by the ones the machine thinks are solvable first, and then the machine will just show you what it thinks is the best solution. And you say, yeah, that's a great solution. That isn't a good one. That is a great solution. And that's how you handle it. And maybe you just read on the first 8 genes of all of them and clear 90% of your pile, okay? So these are the kind of tools we're building. This is how artificial intelligence is disrupting this field of diagnosis. I'll just show you very briefly, right? So we built a tool. We called it Amelie for 2 reasons. One is, I think my guys can put an acronym on anything. So this one is Automatic Mendelian Literature Evaluation. Mendelian is like monogenic. But also, we like this movie, Amelie. I don't know if you guys have seen it. It's a wonderfully charming movie. The tagline is, she'll change your life. And patient diagnosis change patient lives, and that's what we're trying to do for them. So, put a long story short, we have this tool up and alive. You tell me which genes in this first incarnation, you say which genes look suspicious in the patient. You describe the patient's set of phenotypes. So we tell you, okay, you told me your patient has hearing impairment, microcephaly, seizures, hypertrichosis, etc. And here is the list of genes that you suspect, right? So this is what a human expert would now sit and read on each one of these genes. When you hit submit on our tool, this is what you get. The system tells you, I think the causal gene in your case is this gene called KMT2A. And if it's not KMT2A, you should look at this gene second. And if it's not this one, you should look at this DNA I one, right? We've ordered these things. Now, why do I think KMT2A is the gene that is a diagnosis for your patient? Because this paper, Advanced Bone Age, et cetera, is the top-ranking paper from the entire corpus of literature I've read for you. And if you ask me to look at that paper for you, if you just click that link, on the right side, you see the actual paper, so you can go and read that. On the left side, I tell you, look, you told me your patient has renal tubular acidosis. In the paper, I see related terms, abnormality of the kidney, acidosis. You told me your patient suffers from talipes equinovarus. In the patient, I see hip dislocation. So in the paper, I see. So we match what we see in the paper to what we see in the patient, in the hope that diagnosis would be as quick as possible, right? You just put that in front of the expert says, check, looks at all the other genes to make sure that there's no surprises there, and you have a 1 hour, 2 hour diagnosis instead of a whole work week for the human expert. All right, there's an API for it, so people can, integrate into their own tools. There's a standalone version for people who want to run it just internally. So this tool is getting a lot of traction today. All right, so let me show you one last wonderful, terrible fact about genetic diagnosis, okay? So genome sequencing is special. We can get your genome today, look at all of it, even if we don't understand all of it, and there are many things about the genome you should remember from what I told you before that we still don't understand. It's an immutable test in our patients. And like I said, we don't understand everything. So we actually showed, in a new disease is diagnosed almost every day. Hundreds of new diagnoses join the medical literature every year. So it turns out we were first to show that, and a dozen groups have been replicating it since. If you take a negative exome from a year ago, and all you do is you try to solve it again, you don't call the patient back, you just run on the same data again, a year later, you solve another 10% of cases. That's wonderful, right? Because you suddenly appreciate that a gene we didn't know anything about before is the gene that's doing it for this patient. The data was just published. So this is wonderful. Reanalysis will get you more and more value from the exact same data. Problem is, right, I was going to quit on 1,000 cases a day a minute ago. Now you tell me I have to go through all my backlog of 70% of cases every week and look at what. You cannot do that without the machines, right? The machines are absolutely necessary. All right. Last but not least. So the medical domain is interesting. What do I mean by interesting? So my PhD advisor, right, was wise enough in telling me a long time ago something that got etched in my mind in multiple contexts. He said, Gil, always remember, science is first and foremost a human activity, right? And in the context of science, he was trying to tell me, it's not always about the pursuit of truth. It's a human activity, and I think it's similar for medicine. Doctors like to be in charge of everything, okay? But they need folks like me. You could see why there. I'm even faculty in pediatrics now, right? But I'm not a doctor, right? I'll skip the joke. There was a joke here. I won't tell you that. So we have to integrate computer science into medicine. That leads into interesting culture meets. Let's call it that way, right? And there are issues in those respects with trust and leadership and thinking of the right people. So, hospitals have to learn to trust people like myself to lead these integration efforts for them, which is a learning effort. Funding agencies have some ways. So we're always, tack on is fine. If there's an adult in the room, somebody like me could practice. But, we don't feel like we need these adults in the room with us. If anything, we feel like it's the other way around, that they need us. So we try to tell them, look, there's a missing link between an MD and their IT department. You're like a first-time homeowner. You're trying to build a house. You've never built a house before. You go to a contractor and you tell them, build these things for me, right? But you hang a sign on your property, no architect should apply. That usually leads to very interesting buildings, right? If you see where I'm coming from. This discussion is an interesting discussion. It's getting easier over time, but really I think clinical bioinformatics, whatever you want to call it, will become an established discipline, and the people doing it will gradually move from the kitchen to the dinner table. Today they're we're mostly at the kitchen. And this comes along with all kinds of things. I'm asked to review the mission statements of genome centers, right? You take a hospital, you add a genome center to it. So the mission statement, the primary mission statement of a genome center is not to hold the patient's hands. There's a whole hospital establishment that is doing that already. What is the primary goal of a genome center? It's to diagnose the patient's genome. But you find yourself with people, very established people, who call the shots that you have to explain this to them. This is interesting. Anyway, for this goal, two things I'll mention. I think maybe I should have phrased it this way. These inefficiencies in the system are wonderful opportunities to make a change, right? And to gain from that change. So computer science is paramount. And academic ties, that's another thing that I see slightly lacking, are also paramount. Why is that? Because genomics is going to be in R&D for decades. I showed you how we can solve the one-gene diseases. We're making incursions into two-gene diseases, some understanding of more complex. Genomics is going to be both extremely valuable. Remember I told you this half empty, half full glass. We're already extremely valuable. It just will keep getting valuable. More and more. It's going to stay in R&D mode for decades. And while industry excels in execution, R&D is the space for academia. So last but not least, ho-hum. Let me just mention this briefly. Okay, patient privacy. And I think I knew we were going to get tired when we get there. So I'll just mention it, right? Last but not least with a question mark. Okay, so if you kind of saw what I showed you, the best way we understand genomes today is by comparing a patient genome to other patient genomes and to healthy people's genomes. We share all these genomes together to understand what makes somebody sick and what is just a variant in a healthy individual. Genomes only make sense when you compare them to each other. Okay? Let me just skip the example. The problem is, if I share my genome with you, I am subjecting myself to discrimination, myself and my next of kin, because my genome tells a lot about me. It doesn't just tell everything we know now how to read about me. We keep understanding it better, so it tells a lot more about me than we even understand today. And if I put my genome in the public domain, or just give it, to Google, to a hospital, I could get in serious trouble, right? I won't be insured. I won't be hired. So so there are many, many issues there with patient privacy. I just want to make this point for you that it's actually not the case. Okay, so we've recently shown, I won't go through any of the details, we've recently shown in a paper in a Science that you're welcome to pull up if you like, and multiple startups are now getting into that space with us, that you can diagnose patients without sharing any information. There are wonderful tools in computer science that you can use. Essentially, the patients trade secrets between themselves. There's boxes and keys. You play this game. At the end of the game, you know exactly what is the causal gene, and nobody has shared anything with anybody. So the bottom line with that, not with other patients, and not with the likes, the companies who run this thing. A company can run the whole assay for you, and nobody shares any input. That's this awesome thing that comes from cryptography. Compute together, get the exact output together without sharing your inputs. This is where this paper came from. Okay? So we can now, and I won't tell you that unless I doubt you'll ask me. But it's just too late. But keys and boxes. So if we wanted, we could protect patient privacy tomorrow. The question is, do we want to? Right? On the internet, that battle is effectively lost. Right? We get things for free in return for absolutely zero privacy. But on the internet, you cannot, if you go to certain websites or you search for certain keywords, you chose to do it, knowing that it's all getting tracked all the time. Your genome is the gift you got for life. You didn't choose it, and it tells a lot more about you. So the choice is there. The science to make it happen is there. Whether we as a society choose to do it is something else. All right, let me wrap. So I try to give you some taste of some aspect of genomic medicine 10 years hence, how things like artificial intelligence are disrupting it, how the disruption order is logically the right thing, but perhaps not what everybody had wished for to be the right thing. How we're all probably going to be sequenced at birth very, very soon as we get good enough computer code that will only tell us what is useful about our genome at any point in time, because the genome is something like a Pandora box. You do not want to open a Pandora box, right? But if there's a computer code on top of the box, only pulling out the things that you really want to know and you really want to treat now, this, I think, is the future. Artificial intelligence in medicine, computer science at the hospital, talked about culture and leadership. So I'll just start by saying there are huge gains to be made in these disruptive new technologies. A diagnosis for the patients, a hope for a cure for more and more and more, an absolute cure. Remember that key with the stick? Cure. Vaccines coming from the genome. And there are also, with every potent new technology, there are huge challenges. So I'll just close with that so we think about those as well. Discrimination based on my genome, R&D costs for these cures, access to the cures when they come around, all kinds of other stuff. Okay, with that, let me wrap up. I'll just thank, first and foremost, John Bernstein, who's Chief of Medical Genetics at Stanford Hospital, who's been a wonderful collaborator for several years now. The Amelie tool I showed you was pioneered by Johannes Birgmeier and the group, multiple other people in the lab, including Aaron Wenger, a Notre Dame alumnus, and others in the lab, the patients, their families, many others in our division, our department. I'll just put on more names. Max from Santa Cruz, many, many other people, funding agencies, etc. And I will thank you. And if you are not exhausted, I will take any questions. Thank you.
It's a great lecture. You mentioned the human genome, about $1,000 cost now. What would you guess the cost will get down to in 5 or 10 years?
Yeah, so just for sequencing the genome, I think, so this is not my area of expertise. I would cautiously say between $100 to fewer than $100. The push to do it is so large And I don't see any fundamental barrier to doing it that one of the technologies would likely mature enough that, let's say, it would be $100. Right? So when it costs $100, the only question is, do I open this Pandora box or not? It's not whether I can get it. But also, a genome in itself is an inert object. It's just a bunch of A, C, Gs, and Ts like I had on my first slide, right? Maybe I'll put that last slide up. You have to interpret it. These are the tools we're building now. Otherwise, just a bunch of A, C, Gs, and Ts. You can sit on your laptops, type A, C, G, and T, and you have something. So we have to know what to do with it.
Thank you so much for talking to us. So I have two questions. The first is, when you're talking about comparing damaging looking genomes with healthy ones, from my understanding, each one of us is unique, so each one of their genome is pretty unique. So how are you even trying to isolate what is exactly causing it? And my second question would be, let's put the human effort aside. Do we even have the computational infrastructure to scan or to map the genome of the 7 billion population?
Okay, the second answer, let me start with the second question. The answer is, yeah, pretty much. I mean, I exaggerate a bit, but it's just, we have solved the fundamental challenge. All we need is to buy more of these machines and to argue successfully enough with, for example, the insurance that covers the cost that they're actually gaining rather than losing by this endeavor. So for the second question, I know that we can get all 7 million kids that are born every year now, but we can get hundreds of thousands, I think, with infrastructure we have now. That is the second question. So the answer to the first question is slightly more technical, and these are like the things that I thought would be slightly less relevant for the talk today, because there were so many other things I wanted to cover with you guys. So let's put it this way. It is literally as I said it is. If you show me 60,000 genomes of people who do not have a severe genetic disease, okay? They were born healthy babies, most of the population. Show me 60,000 genomes like that, and show me here, I'll show you the example. Let me briefly go to that, because it's just, I think, around the corner here. The thing I skipped through. If you show me 4 patients that look similar to each other, they're just, they all fall on this category of Miller syndrome. They really would all have something in them that you don't see in any healthy individual. That seems like a bad idea. And if you look at something that we know about the gene that they're messing up, what we know about the gene kind of makes sense in the context of their disease. Like, that gene is involved in some operation in the liver. Sure enough, that is a liver disease, and you found what does it. So without going into too much technical details, we have now used that logic 5,000 times and successfully pulled out the mutation. All you have to do is, sit through like one lecture in genetics proper and see how that is done. If you combine the semantics of what we understand of the genome code today, plus the statistics of what patients have versus what controls have, you just get it. All right. Yeah.
Yes. In curing the bubble boy, for example, his immune system, did the researchers use deuterium depleted water, which costs $300 a gallon, or what is their preferred type of water in conducting these very delicate procedures? And I have a second question. Are telomeres, the length of telomeres being studied at Stanford in relation to slowing aging?
All right. The answer to both questions I think, is easy on my part. The answer to the first question is, I don't know. I do the informatics, and I am not privy to the specifics of the procedure, and I wasn't involved in that particular curing process. So I could look it up, we could look it up, but I don't know. And the answer to the second question is pretty similar. I suspect people at Stanford are studying telomeres. I know there are people at Stanford who study aging. Typically, telomeres are interested in the context of aging. This is not what I do, and it's not what I was talking about today. Hopefully that's helpful. But that, we should always profess the limitations of our own knowledge. So, all right. Somebody up there.
I just had a question. You talked about science is getting to a point where they can pull out mutations, like the bubble boy you talked about. Could you talk about the science of possibly replacing like microdeletions?
Conceptually, it's the same thing, okay? There is nothing conceptually different about fixing one letter versus changing a few letters. There's nothing at the level that we are discussing today. If anything, it's actually the case that our tools are slightly more blunt right now than I described them. So it's easier to deal with micro deletions to some extent than it is to replace a single base pair because our hammers and chisels are a bit rough. But at the conceptual level here, we are going to be able to do all of them. People are figuring out, trying different combinations of reagents. All of that is going to happen. Let me tell you guys one last thing that I think I didn't put in the slide, just for fun. If you're interested in artificial intelligence in any context, right? A big disruptor right now. I'll tell you what I tell the AI students who come to Stanford when they first come in even at interview week. I tell them, look, tongue-in-cheek, but there's a grain of truth in there. I tell them, look, there's only two types of AI researchers in the world right now. Those who work on healthcare data and those who haven't worked on healthcare data yet. Okay, so healthcare is just producing so much data. It's becoming huge in the context of AI, and I think a lot more of the AI workforce and a lot more of the innovation disruption is moving there. That's for you to consider.
We do have time for one final question from the audience. Well, if you could all join me then in thanking our speaker for today, Professor Gill Bergeron.